questions and a chat. So maybe next slide on, please. So just to say, this series of online events is part of a wider European Union programme, which is to engage citizens across 14 countries around SDG 11, which is creating resilient, sustainable communities, something I think is more important, particularly in recent COVID times. So today's webinar, and maybe John, if you go to slide 2. Today's webinar is around the ESAI Postgraduate of the Year, which is something that we will explain a little bit more in a second, but we're delighted to welcome a current postgraduate winner of the year and a former, to provide a platform for them to share their work. So maybe the first is Philip Shine. Philip won in 2017, if I'm correct. And maybe just to check, Philip, shout out, are you there? Maybe you can unmute and just say a quick hello to let us know.
Unshot.
Good. So Philip's going to be talking about his experience with the postgraduate year and sharing some of his research work. So Philip was working with Cork Institute of Technology, CIT, and he was looking at electricity use in Irish dairy farms, a national approach to improving efficiency using artificial intelligence. And again, delighted to welcome our current winner of the year, Molly Wiliams. Molly was doing her research out in the Water Institute in DCU, who are going to host Environ in October. So Molly, just a quick shout out to the universe there. Are you ready? Are you out there?
I am ready. Hi, everyone.
Excellent. Welcome, Molly. So Molly's going to be talking about her research, which is based around novel methods of monitoring Atlantic salmon using environmental DNA. So just before we start, and I hand over to the guys to just a quick word on the postgraduate competition and how it works. So this role of the ESAI is to encourage, to support, but also to recognise environmental research and to recognise excellence in environmental research. So the Postgraduate Research of the Year was an award started back in 2013, and it's open to PhD or master's students. And any of you out there, I would strongly encourage you to apply. It opened in April, will be closing end of September. And the winning entrant receives a prize of €500. Money's always a great motivator, but more importantly, it gives you an opportunity to disseminate your research to a much wider community. We give you an opportunity through events like this, through our Environ conference, where you're invited to make a presentation, and through our websites. So for any early stage researchers out there, it's a great opportunity to really kickstart and get your research to a wider community. And just briefly, we judge it on 3 criteria: scientific excellence and originality, impact across a broad spectrum of sectors, and dissemination, communication, how you communicate your material. So maybe, John, just the next slide. So as I said, we would encourage any of you thinking to apply. And one of the criteria, which is increasingly important, and in the recent COVID outbreaks, the importance of disseminating your research to a broad community, both technical and non-technical. Just ask Dr. Fauci about that one. So this is part of how we judge is how you disseminate your research throughout your postgrad or master's program, and how you explain the impact and how you look at opportunities to engage with wider communities. So as I said, the competition is currently open. There is a link on our website on how to apply. So we would encourage anyone thinking about it or to share it with your colleagues out there. It is a great opportunity. So with no further ado, I'll hand over now to Philip. And Philip is, as I said, won the Postgraduate Researcher of the Year in 2017, if I'm correct. And Philip has been working on the board of the ESAI since. and has been working, doing great work. And Philip's going to talk about something that is very topical, the whole issue of farming, of resource use, of improved efficiency. So Philip, the floor is yours.
Spot on. Thanks a million for that, Liam. Thanks for that introduction and overview of the postgraduate researcher of the year competition. Just to expand on that introduction a little. So currently I am a second year postdoc in CIT, having finished my own PhD here in 2018. So during my PhD, I was part of a team that were awarded funding from the ESAI RD&D fund to develop a decision support system for energy consumption on dairy farms. And this work really complemented my own PhD at the time, which focused more towards the application of machine learning algorithms to predict dairy farm electricity consumption, in comparison to that of the decision support system, which is more based around mechanistic modelling. With those mechanistic models integrated with various renewable energy technology models to allow users to assess the feasibility of certain energy technologies. So what I really found during my own research was that these machine learning models, they had the dual benefit of offering highly accurate predictions, while at the same time requiring far less input variables compared to the state-of-the-art mechanistic models. And these variables could really be easily collected on a large scale without the use of specialized equipment. So since completing my PhD in 2018, we've been awarded further funding to further develop the decision support system, while also being able to realise the work from my own PhD to develop a closed loop system for ensuring the continual development of energy consumption on dairy farms over the next number of years. So just to summarise what I have in store for you this evening, first I just want to go through a remote monitoring system that we have installed at the moment that is installed and maintained by Chagisk, and this really forms the basis for all of our work going forward, because it allows us to identify usage trends in energy consumption over time to ensure that any future models are kept up to date with those usage trends. And this feeds directly into our decision support tool. And this tool is it's an open source web application that allows farmers, researchers or policymakers access to scientific outcomes over the past number of years in relation to investments in energy efficient and renewable energy technologies specific to dairy farms. Or it's also a tool for dairy stakeholders looking for information regarding their agri-energy consumption on beef and sheep farms, for instance, as well. And lastly, just is the development of our National Artificial Intelligent Dairy Energy Application, or NADIA, as it is most commonly known as these days. And this aims to integrate national level survey information with those machine learning models that I was able to develop over the course of my own PhD, that are validated using data collected as part of that remote monitoring system that I mentioned earlier. So I'll go through this in greater detail and how these three sections all kind of tie into each other over the next 10-15 minutes or so. But before I delve into that, I just want to touch briefly on the Irish dairy industry to just give you some background to why we're bothering with this work at all. So at the moment, there is in around 18,000 dairy farms in Ireland, but it's forecasted really that this number will actually decrease over the next number of years as the milk production per farm increases. So we'll see a much more intensive milk production system. And we've already seen this since the abolishment of milking quotas in 2015, whereby we've seen an increase in milk production of 25%. And this is all well and good. It obviously has some positives associated with it, in particular when we look at the value of our dairy exports. However, there are also some negative impacts of that must be addressed. Firstly, when we look at the increased milk price volatility, so the income received per litre of milk by dairy farmers is much more unreliable. And secondly, then, with increased milk production, inherently you're going to have an increased consumption of energy and emission of greenhouse gases. And we all know that methane production from dairy cows is a major issue, particularly in Ireland. So the agricultural sector is really under pressure to improve its overall sustainability. So just to summarise what's going on at the moment and where we're coming from, Farmers are now producing greater volumes of milk, so they're working harder, they're putting in longer hours, but they're receiving less financial rewards at the same time. And they're also required to produce less greenhouse gases. So no immediate solution really is available to reduce those greenhouse gases from dairy cows. However, one such option is to optimize the energy production on those farms, which is really where my work comes into play. So the following slide just gives us an overview sense of the remote monitoring system we have installed on over 60 farms, with their electricity consumption being remotely sub-metered via 3G network. In addition, with infrastructural and managerial survey information, stock data from the Irish Cattle Breeding Federation, milk production data from the individual milk processors, as well as environmental data provided by Met Éireann. So it's all stored and continuously updated in a cloud-based database. And this database allows us to quantify usage trends and changes over time, as well as to quantify different relationships between farm equipment, managerial processes and electricity consumption. And what we found was that during the day, electricity use is distinctly bimodal, and you can see that just in the graph up in the top right hand corner of your screen, whereby a clear spike in consumption can be seen during and after each of the 2 milkings carried out each day. And when we further broke down this consumption into its constituent parts, what we found was that in around 50% of consumption was directly related to the milking process. So from this, the hypothesis was that energy or electricity consumption on dairy farms had high potential to be predicted. And these findings fueled the development of the mechanistic model for electricity consumption on dairy farms, which was developed and published in 2014 to a high degree of accuracy. And this really formed the basis for our decision support tool. This tool is an open source web application which aims to reduce the carbon footprint of Ireland's dairy farms through informed decision making. And it's available just at the link in the top right hand corner of your screen. What it does is it allows users to quantify the monetary, electrical or environmental impact of producing milk and assessing the applicability of various renewable and energy efficient technologies. And really increasing the uptake of these technologies on dairy farms has a dual impact of firstly reducing the required load from the electrical grid, and secondly improving the penetration of renewable energy on Ireland's grid. And this will have a positive impact on Ireland achieving its EU 2030 energy targets. And most importantly, what this tool aims to do is to achieve these targets in a means that is as cost effective as possible to the dairy farmer, ensuring that their decisions that they're making related to their investments are informed. Because typically electricity cost savings that are described by technology suppliers can't really be relied upon, as these figures are typically calculated for the average dairy farm. So there's a high risk of greater long-term running costs for dairy farmers if these pieces of equipment are not managed and sized correctly. And lastly, just on the decision support tool, it also offers a means for policymakers looking for decision support in related to their decision making, helping them to identify those technologies that are worthwhile to support with grants, and also to calculate a suitable level of funding or grant aid required to meet certain energy targets. So this tool has 5 key components. The first one being that mathematical model I mentioned a couple of slides ago. What it is really is it's a mathematical representation of the 7 main infrastructural processes on a dairy farm. So you have your milk cooling, your milk harvesting, water heating, pumping, etc. And the second component then is our energy models. So we have 6 energy efficient and renewable energy technology models, each of which have been validated using infrastructural equipment either in CAT or the energy labs in Teagasc Moor Park. The third component then is just analysis functionality built into the tool, which allows for monetary variables within calculations to be adjusted according to unique user-specific conditions. These include variable cost structures, with various levels of grant aid, investment cost, availability of feed-in tariff, etc. And next is inputted optimization functionality, and this allows users to automatically identify optimum solar PV system sizes, milking times, water heating systems, or water heating system start times that will either maximise the return on investment or minimise carbon emissions on a farm-to-farm basis. And the last component then is a couple of data aggregation sections, which provide summary of data collected on beef, sheep and dairy farms. This just provides an outlet for users looking for further information on the usage, the cost or energy related CO2 emissions within these agri sectors. So as I said, this tool is actually deployed at the minute and it's available via the link at the bottom of your screen. So I've just demonstrated quickly here how easy it is for a user to actually generate outputs using this tool. So the first step really is the user wants to input information specific to their own farm conditions. And this just looks for pieces of information such as farm location, herd size, their milk cooling system, water heating system, and it's all variables really which have a major influence on that electricity consumption trend that we saw earlier. And the user just inputs these farm data details into the tool and then moves on to inputting details related to the energy technology. So there is 5 potential technologies mentioned here. We've variable speed drive, we've heat recovery, we've solar water heating, solar PV systems and wind turbines. So for this example, the user has inputted details related to a solar PV system with a system size of 5 kilowatt peak at €1,200 per kilowatt peak with 60% level of grant date available. So the user then just runs the tool and gets the following output dashboard. So you can see here in the top right hand corner that using those specific farm conditions with those technology conditions, this system will pay back in just over 4 years with a net investment cost of €2,400. And as well in this dashboard, you can also see pieces of information such as return on investment, dairy farm energy use, the energy generated by the PV system, the carbon saved over 20 years, and the cost per tonne of CO2 reduction saved. So I haven't really delved too much into this tool for fear of time really. But I suppose it's worth noting that this system is readily available at the moment. However, over the past couple of years when we have been developing this tool, we did kind of notice that it relies solely on the farmer's own initiative to look up this tool and to locate information related to their own energy investments. So we looked at how this barrier could be removed, and that really fueled the development of our National Artificial Intelligent Dairy Energy Application, or NADIA. And this really closes the gap between Ireland's dairy farmers and access to this decision support tool. And really, it does this by combining the national level survey information, which is collected by Borbia's Sustainable Dairy Assurance Scheme, and those machine learning models that I created over the course of my own PhD. So NADIA will allow Borbia or other government bodies to generate dairy energy related reports on Ireland's population of dairy farms. But what it will do, and what's kind of cute about this system, is that it will also allow Borbia to identify those farms that are using their energy consumption inefficiently, so that it allows that intervention to be carried out, that Borbia can then direct those inefficient farms towards that decision support tool, or it could directly offer those inefficient farms their own unique incentives or levels of grant aid that will help them to improve their energy sustainability over time. And this system really is only really possible due to the nature of those machine learning algorithms, which have the ability to generate accurate predictions using relatively coarse input variables that can easily be collected over a large scale without the use of specialized equipment. So again, I won't get into the specifics of the machine learning models themselves, but it's worth noting that various data mining methods were used, such as model selection. So we've assessed various models such as neural networks, support vector machines, random forests, as well as other methods such as feature selection, hyperparameter tuning, cross-validation, really with the end aim in order to generate a model that generalizes well over a population of 18,000 dairy farms using a specific cohort of 60 dairy farms in this case. that we have direct access to. So if anyone is interested on those specifics, feel free to give me an email after the presentation, and I can provide you with a bit more detail. So the following infographic just summarizes NADIA again, really, and just provides an overview of how that data is actually constructed. So we can see that Borbia, through their Origin Green project, they collect met data from Met Éireann, national survey data as part of their dairy assurance scheme, and also data from Teagasc and additional stakeholders that are required to be inputted into NADIA to generate those reports. And in the meantime, then we have those machine learning models that are being generated and continually trained over time through that remote monitoring system that keeps NADIA up to date with current usage trends that are going on at the time. And this allows for the generation of national or farm level reports and allows Borbia to identify those farms and to direct them towards the decision support system, and possibly offer them certain levels of grant aid that will ensure their continual improvement of their energy systems. So this project began at the start of this year. As I mentioned, it is a continuation of the work I put in over the course of my own PhD. So it's really rewarding to see it actually being realised. So just to tie things up here, a vast majority of research and development has been gone into energy consumption over the past decade, when we're really only seeing the fruits of that labour over the past couple of years, with the announcement of numerous dairy energy related grants, which are kind of well timed with the deployment of our own decision support system. And we have been getting really positive reviews from that decision support tool since its deployment in 2019, and which is really good to hear, because we did place a specific focus on the specific user experience, because we were quite cognizant of the wide range of users that could be using the tool. So we wanted to keep everyone happy, for want of a better word, and this was achieved through holding various focus groups and various open days over the course of its development. So it was continually developed over a number of years. And just lastly, this work is currently ongoing in relation to development of NADIA. As I said, it only really began at the start of this year, and it has an expected delivery of early 2022. So all going to plan, I should be able to delve into this a bit more at Environ later on this autumn. But for now, I suppose that's all from me anyway. If you have any questions, feel free to throw them into the chat, and I'll do my best to answer them as best as I can. So back to you, Liam, I suppose. Thanks very much.
Thanks, Philip. Excellent presentation and overview, and I think a good indication of kind of the standards involved in the postgraduate researcher of the year. We will have time for some questions after Molly's talk also. So if anyone has some questions, they can post them in the chat, raise their hand, or we'll have some time after Molly's talk also. I just have one, Philip.
Yeah, fire on.
Well, two quick ones. I suppose, do you think farmers can transition from energy consumers to energy producers from your work?
I think it definitely has a place. One of the things in relation to energy consumption is that the energy prices that we pay when we're purchasing energy from the grid, it's susceptible to increases, and it looks like it will be continuing to increase over the next number of years. So having their own independent source of electricity, even if it is 50–60% of their total electricity is generated independently, that could have major factor on their production cost over the next number of years. And that impact will only increase as the electricity that they're paying from the grid actually increases in price. So in terms of them being able to transition, I do think that we are on the right path in order for that to be the case. I know solar PV systems have recently been added to government grants, whereby farmers can receive a 40 to 60% grant towards their solar PV system. So those little initiatives make all the difference when it comes to looking at that return on investment and looking at that payback period. Farmers really do want to see that they are getting the best bang for their buck, and that's where we're hoping that we can help with our decision support system.
Okay, good. And it's maybe a difficult question, but it has to be asked. A number of weeks ago, at one of the ESAI webinars hosted by DCU, we had John Gibbons. And John is a big proponent of a revolution in farming and a new Green Deal with nature. And I suppose it is kind of the elephant in the room, where you kind of described there a smaller number of highly efficient farms. So I know, like, it's a little bit outside of what you're specifically doing there, but it's in the role. Like, how do you square that kind of circle where your demand for increased efficiency, farming is a business, so it has to be cost effective, but yet on the other hand, we have climate change, we have carbon emissions, we have Lancet articles talking about revolution in how we produce food. Just wondering, you know, it's kind of a general question, and like I said, maybe a little bit unfair, but what's your thoughts around that whole discussion?
Yeah, thanks for that, Liam. Yeah, I suppose dairy farming, it will also always have, or it will have a part to play in Irish society, whether we like it or not. It is just a case of trying to put that box around those farms that are producing their milk or producing their beef as efficiently as possible and getting that balance right. But to answer your question, I think I'd really need to sit down and have a proper think about that if I was to give any answer that I'd be able to put my back into, I'm afraid. No,
that's fair enough, Philip. Probably something best discussed over a pint.
Yeah, exactly.
We do have a question from Arno Frick. Philip, have you ever looked at applying the system across the globe, for example, in countries where the dairy economy looks slightly different?
It has been on the radar, and energy consumption on dairy farms has been carried out, like in the States. I know Iran and Italy also are very focused on energy consumption on dairy farms. That is on the radar, and that data has been collected. It's just a case of, I suppose, putting the correct collaboration in place. It's definitely possible, but I suppose at the moment we're focused on showing what this kind of technology can actually output and what benefit it has before going to those countries and saying, here, look what we've been able to achieve with this system. You can also look at this as well. We can help you out. You know, so it's just about getting that international collaboration together. But the data is there, and the methods are hugely the same, which would make it that bit easier.
Okay, good stuff. And just one more quick one before we hand over to Molly. We have a question from Gulan. You say energy, electricity, but do you take off farm energy consumption, for example, for mineral fertilizer production?
Yeah, good question. So my work is specifically related to electricity consumption. So electricity consumption is about 60% of total direct energy consumption. And really the reason we focus on electricity consumption is because it can be directly controlled by the farmer in terms of infrastructural changes that they can make. So yeah, my research just focused on electricity consumption.
Okay, excellent. And then just the last quick one, just in general around the Postgraduate Researcher of the Year, what would you say to anyone tuning in through the webinar series who's kind of thinking about applying but a little bit reluctant or a little bit scared by the whole process?
Yeah, I can't overstate it enough how beneficial it was for me. I remember back when I was doing my PhD, I was able to even just look at the application, look at the three pillars, the societal, the economic and environmental impact. And even 12 months before I was able to apply, I was able to look at the application, and I kind of just realized myself, I was like, right, maybe I'm not ready for this. I look at it again in 12 years, or sorry, 12 months. And over the course of those 12 months, then it just gave me a chance to think about my research in a bit more detail and how it can impact these three categories. So it really gives you the opportunity, or gave me the opportunity to kind of almost tailor my work towards like having a greater environmental impact, a greater societal impact, and greater economic impact. So that actually when it did come the time to apply, I knew exactly what my research was about, and that just gave me a much more stronger PhD as a whole when it came to submitting for my thesis. So really I can't overestimate how important it was for development of my PhD. Even just looking at the application and seeing what it's about, it just gave me the opportunity to look at my research through a different lens, for want of a better word. So if you are thinking of applying, definitely go and have a look. If you think that your research, it mightn't be mature enough, have a look at the application anyway. In 12 months' time, that could be completely different.
Okay, great stuff, Philip. Thanks very much. And if anyone has any more questions, they can post them in the chat, or also Philip has his email up there, or he can be contacted through the ESAI also. Great stuff, Philip. So maybe now we'll hand up to Molly.
Yeah, no worries.
Molly, are you out there in the universe somewhere?
Sorry, yeah, I am here. Can people see my screen?
Yes, that's good, Molly. So Molly's our current 2019 postgraduate winner of the year. I believe Molly's also been doing some interesting work, COVID related as well. We might chat about that later. So Molly, over to you if you want to introduce your research yourself and maybe take us through your own experiences.
Yeah, perfect. So just hi everyone. So I'm actually currently still a PhD student at Dublin City University. I applied for this award in my third year, and which was great. And I kind of am in a fortunate position, but I'd agree with everything Philip was saying there in kind of terms of kind of looking through the design and everything of what was needed and kind of pitching, even just thinking about your PhD slightly differently around the kind of criteria. So what I've been focusing on then is developing novel methods for monitoring Atlantic salmon using something called environmental DNA. So hopefully some of it's a bit technical. Hopefully we'll all get through it by the end. And yeah, then you can ask me questions or contact me after the webinar if need be. So just a brief kind of background into me and my story. I'm obviously English, as you can tell from my accent. I started my undergrad at the University of Warwick doing a BSc in biological sciences, and I was fortunate enough to carry out 3 kind of major research projects whilst I was there in several different labs that got me interested in environmental sciences. So they were identifying marine bacteria, which were degrading DMSO, or not in the actual case of my project, looking at kind of ancient barley and how barley has evolved over time, and then identifying bat species from guano that might be left in attics or anything like that. And this kind of got me thinking about DNA in the environment, even from a kind of second year undergraduate student when I started those projects. I then moved to King's College London to do my master's in forensic science, so entirely different. But as part of that, I was fortunate to come over to DCU Water Institute and carry out a project on microplastics in Dublin's freshwater, marine water and biota. And whilst I was there doing that project, the kind of PhD opportunity came up and I went for it, and I'm now, about to start my 4th year, I guess, of my PhD in the School of Biotechnology at DCU, investigating the development of a novel biosensor for detecting single species from environmental DNA. So So I'm going to focus then obviously on my PhD work. So I'm sure everyone here probably knows that biodiversity and conservation are hugely important for human health, food security, everything like that. But the numbers are quite, well, very concerning. So the World Wildlife Fund reported, this is the 2018 report, a global decline of 60% in population sizes since 1970. But when we move that just to fresh water, this decline actually rises to 83%, which is hugely concerning for our biodiversity globally. So we know then, in order to try and conserve the biodiversity we have to be able to monitor what's already there. So we can focus then on endangered and invasive species, but then also a community level of what we might have present in our environment. The problem then with kind of the traditional methods used, or the reason that my research kind of came is because traditional methods such as our commercial catch, wild catch, traps and campers, wire tagging, you can read, electrofishing, all of these methods that are great and they have their place and they are still used, and I'm sure some of you probably can tell you way more about these than I can. They're often quite invasive. They require direct capture of the organisms of interest. So be that through trapping or electrofishing, and even though we're releasing them again, we have to have a direct interaction with the organisms that we're trying to study or just simply count. And this is great, it has its place, but it's also problematic if we're going to try and conserve these organisms, because we ideally don't want to interact with them. We kind of want to be as far removed from their natural environment as we possibly can, while still being able to monitor them and protect them. So there's kind of several other problems then with our traditional techniques. They can be, as I said, potentially harmful to the organism. They are also quite disruptive of the habitat. So if we're to put in big traps, this can be hugely disruptive to that habitat. And they can also be quite expensive and labour intensive. So that's kind of the drive then behind environmental DNA as a concept. And I'll explain to you a bit more about what that actually is. But the main kind of reasons that it's kind of exploded in the molecular ecology world is because it's a non-invasive tool. So we can effectively do direct monitoring of species straight from the environment without having to engage with the organisms of interest at all. It's also, in theory, cheaper and less time consuming in the long run once the assays are kind of fully optimized and up and running, and can be used as both a single species monitoring system and as well as a biodiversity assessment system. So what is environmental DNA then? I've said this word multiple times already. We define it then as genetic material obtained directly from an environmental sample. So this can be water, soil, sediment, ice, cores, whatever your environmental kind of source might be, we can probably get DNA from it. And this can come from multiple sources from the organism itself. So damaged tissue, skin, scales, it could be free DNA or metabolic waste as listed here. So in the same way, you might know for yourself, we shed our skin constantly into the environment. Our organisms do this too. So our freshwater species are also constantly shedding this DNA into the environment. And this is a great source for us to be able to tell you what's there in an entirely kind of removed way from the organism itself. So we can analyse environmental DNA then in kind of two main ways. Everything sort of starts the same. This is a generalisation of what we might do, and it's my method. There are slight variations in this. But you'd go to your sampling site, or hopefully multiple sampling sites. You can take your water sample, and then we commonly would filter this water sample and trap our DNA then on that filter. This is when our path then separates depending on what you're interested in. So depending on whether you are looking for single species or for biodiversity, so more of a community level assessment. So if we're to look down our single species pathway then on the left, we extract that DNA from the filter. We can then use specific primers that are species specific to amplify just our target's DNA, and we can then detect that with specific probes. So this would be our kind of conventional single species environmental DNA qPCR technique, and that's what's done very routinely in the field now. We then have our second approach, which is still developing and isn't quite as refined yet as our single species, and this would be our metabarcoding. So we still amplify our DNA, but this time we use non-specific primers. We can then do high throughput sequencing of our amplified products to kind of get a picture of everything that's there. And then it's a big bit of bioinformatic job of assigning the taxonomy of all of the sequences we have there. And the huge kind of downside with that at the moment is that the reference databases just aren't there. So we might get a kind of indication of what biodiversity we have, but without the reference sequences to compare our kind of high throughput sequencing to, it's quite hard to actually fully get the full kind of picture of what might be in our environment. What I'm going to focus on today then is single species. This is where my research comes into it. I'm not looking at metabarcoding currently with my PhD. So why use qPCR? You can see this is only 15 papers, but there's a lot of people have developed qPCR assays now for a whole host of species, a whole host of environmental samples. And this is because, like when anyone's using qPCR really, it's a highly sensitive and specific amplification of our target, which we know to be in low abundance in the environment. Obviously, our water sample can be quite large, especially if you're looking at, I don't know, a full kind of watershed or something like that. You have a large area, a lot of moving parts, a lot of factors to take into play and to consider even. And we know then that the DNA amongst that kind of water sample might be fairly lowly abundant, but qPCR allows us to still detect that because it is a highly sensitive method. It does, however, have its problems. So the main limitations that kind of concern me, and the reason I've come up with the new method that I'm going to describe to you in a bit, is based on these kind of four main factors. So the first one is to do with susceptibility to inhibitors. So in the environment, we know can contain a huge amount of inhibitors that we know to affect qPCR. And this is a huge issue when we consider false negatives, especially if we're trying to detect invasive species, because we want to know if they're there. And if we have high numbers of inhibitors in our environmental sample, we might get false negative results back. qPCR then is also fairly difficult to adapt to a biosensor, and this is kind of where my research comes in, and this is mainly due to the high temperatures and thermocycling needed for qPCR technology. It can also be difficult then to distinguish closely related species, and this is due to a tolerance of primer mismatches. So what I mean by this is if we're looking at two close relatives, say for my research, I look at Atlantic salmon and its close relatives, brown trout and Arctic char, which are also in the Irish water system. We need to have areas of the genome that are highly different from each other for us to target our PCR assays with enough kind of mismatches between our primers to ensure that we're not getting false positive results from closely related species. qPCR then can also be time consuming and is on the whole limited to a laboratory setting with required trained kind of personnel. And this isn't entirely true. People have been developing on-site qPCR systems, especially from the medical field for point of care testing, but there are a lot of limitations with it, and it can be quite difficult kind of design in terms of making it an on-site system. So that's where my research comes in then. So I have, obviously not on my own, in my team developed a new method of molecular detection of environmental DNA, which couples CRISPR-Cas with an isothermal method of amplification called Recombinase Polymerase Amplification. So this is the overall strategy then. It can be quite confusing when you kind of look at it all together. But effectively, I'm using the exact same extraction method as everyone else. So I'll have my water sample, I'll filter my water and trap the DNA on the filter. I can then extract my DNA from the filter. And then instead of using a PCR-based approach, I am amplifying my DNA using this isothermal method called RPA. And this only takes about 20 minutes, so it's a lot shorter than our PCR. And I can then use a CRISPR-Cas system, which I'll explain as we go along, to detect then our amplified products in a highly specific manner. So I'm going to focus first on RPA. So RPA stands for Recombinase Polymerase Amplification, and this is an isothermal method which effectively uses a recombinase enzyme to form a complex with our primers and promote strand invasion. We can then stabilize this the initial denaturing of our strand where our primers are inserted just using single-stranded binding proteins. And we then use a very unique DNA polymerase, which is able to displace our DNA, our double helix, as it's elongating its target. So unlike PCR, which requires the high temperatures to denature our double strand, we use the activity of the DNA polymerase to actually displace our two strands whilst we're elongating our target. And this makes it a highly efficient system, which allows us to reduce the amount of time to actually carry out the amplification. Because we know from PCR, you have to carry out these cycles of heating and cooling in order to denature and anneal. And as I'm sure you will know, and elongate our target, they're almost 3 separate steps. Whereas with RPA, 2 of the steps are combined into 1. And we remove the temperature aspect altogether. So all of this can actually occur then at 37 degrees, which is fantastic in terms of bringing this technology on site, because it already requires less energy and effectively just 2 heating blocks that heat to 37 rather than having to do this thermocycling. The problem then with RPA, hopefully you can see quite clearly from this gel, is that it's not very specific. So I designed an assay for Atlantic salmon and tried just the RPA in isolation. And you can see then that we get the desired amplicon actually in both Atlantic salmon and in our brown trout, which isn't great. So that's then where our CRISPR part comes into it, which is we stole effectively from the molecular, the medical field. So this is a bit of a complex slide. Don't worry too much. It's just if some of you might never have heard of CRISPR, so hopefully it's just to describe that to you. So CRISPR then stands for Clustered Regularly Interspaced Short Palindromic Repeats, and effectively it's a system that we stole from bacteria. So in several bacteria, they have an ability to insert what we call viral protospaces into their own genome. So these are effectively short strands of viral DNA that they insert into their own genome. And this acts as their adaptive immunity. So they're therefore able to have a memory for viruses over an evolutionary scale, viruses that have previously infected them. And these short protospaces can be transcribed into RNAs, which can then act as a guide for our nuclease, which is our enzyme that actually does the cutting. We then have a very specific sequence, which is what we call a PAM site, and it's effectively a recognition signal. So this PAM site is only present in the viral genome and not in our protospacer. And this allows the bacteria to distinguish their own genome and the insert that they've made against viruses that are actually attacking them. So it's a neat little system that allows us to distinguish these really closely related sequences apart. We can then use what's called a nuclease enzyme, and effectively this is just the molecular scissors of the bacterial system. So they just come along once they've been guided by their RNA, they'll bind to their target and then cleave the target. And this just disables the virus in terms of bacteria. There's lots of different types of nucleases. The one we're interested in is Cas12a. So this isn't the primary one that people are using in genome editing, and that tends to be Cas9. But Cas12a has a really advantageous secondary function. And this is that upon recognition of its target, it has an indiscriminate cleavage activity of single-stranded DNA molecules. And we can utilize this to insert a single-stranded fluorophore molecule, which effectively allows us to detect when the cleavage activity of our target is happening. So hopefully this schematic explains it a little bit better. So we have our target DNA, which is followed by a PAM site, because remember, this allows us to distinguish the viral DNA from our own DNA, so it's hugely important. We then have our Cas12a with the guide. These then bind together to form a complex, and in the case it would cleave the target, but we kind of don't care about that. This complex then, if we insert a fluorofluor quencher molecule, will then cleave our single-stranded molecule, and we can then detect the fluorescence. And really importantly, the Cas12a nuclease won't cleave the single-stranded DNA molecule in the absence of the target. So it has to form this complex in order to have the secondary activity, which is the cleavage of our fluorophore. And again, this occurs at 37 degrees, which is great for me in my research. So what I did then in terms of Atlantic salmon was I targeted a mitochondrial gene and looked at kind of these 4 levels of specificity. So I have my 2 primers, my forward and reverse, which are for the amplification side of things. I then have a unique PAM site that is only present in my Atlantic salmon and is missing from my brown trout. And this allows us to be able to distinguish, well, the Cas enzyme, not us, to distinguish the 2 sequences. And we then have our guide RNA region, which also has as many mismatches as possible. So this just allows us to have 4 levels of specificity with ideally, obviously, as many mismatches as you can possibly get between the sequences. So did this work then? It's all very good in theory. How do we actually test this? So we started then with just recombinant DNA. So I cloned in my 2 kind of insert regions into 2 bacterial kind of plasmids, both for Atlantic salmon and brown trout. And I tested just the Cas12a alone. So we know from the agarose gel that I've previously shown you that the RPA does amplify both. So it was a question of whether the Cas12a can then distinguish the 2 species. And you can hopefully see from the fluorescence that these sequences were different enough, and the absence of our PAM site allowed fluorescence to only be detected in Atlantic salmon and to not be detected at all with our brown trout. And this is obviously using a plasmid, so it's a very high concentration of and low complexity DNA. So this was a really good indicator, but does it work in the environment? You know, it's all well and good when things work in a very controlled system, but our environment isn't that. So we then did an initial pilot study collaborating with UCD and the Marine Institute in the Burrows Hall catchment in Mayo. And we only tried a very small subset of samples, and we've gone on to expand this now kind of after I did this initial study. But we tried 3 samples where we knew we had Atlantic salmon DNA from electrofishing studies, and then 2 samples where we knew we shouldn't have Atlantic salmon. And we actually saw a perfect kind of match to what we expected to see. So in our Burren, Delore and downstream, which downstream Rough River, where we know there's Atlantic salmon presence, we did see high fluorescence kind of signals that were above a background noise. And in our other samples, which were known to be absent, we didn't see it. So this is really, really promising. It made us very excited in our lab and kind of highlights that this system could work for environmental DNA in a very kind of proof of concept kind of study. So if you do want to read more about this study and the design and everything, we did publish this last year in Molecular Ecology Resources, so you can have a look if you're interested. But why might this method improve eDNA detection, and kind of where is my PhD going in terms of this new methodology? So for me, I'm interested in the biosensor. So the isothermal conditions kind of just simplify the development of a portable device. And there's still, hopefully in the last year of my PhD, we'll get to the biosensor stage, but it's still a lot of work on kind of top of this initial molecular kind of technology that we've developed. We're also then hoping it will be great for distinguishing closely related species. So on top of our kind of mismatches in our primers and the guide RNA, which is equivalent to kind of your probe in qPCR, we also then have the necessity of the PAM site. And this just adds that additional level of specificity. So from what we've kind of our initial study so far, it seems to be that if the PAM site isn't present, the Cas12a won't cut and you won't get that secondary activity. So this is kind of our crux then of the design, and hopefully will open up a new way of detecting closely related species. We also think it might be able to work in highly degraded samples. This would rely on the not needing the amplification step. So if the Cas12a could work on its own, which we haven't investigated, there's the potential for detection down to only 30 base pairs of sequence, which is a really small amount of DNA and could be crucial when we're looking at highly degraded samples. We're also then hopeful that it might be able to work where there's high inhibition in our environment. And this is down to that different amplification chemistry. So the literature has already kind of shown some of the known inhibitors for PCR don't seem to inhibit RPA. So hopefully where water contaminants are high, we might be able to use the RPA CRISPR method to still detect our species of interest. Without using qPCR. So obviously they all have their place. And I think from my perspective, I see all of these methods kind of being integrated together depending on your circumstance and depending on your question. So we still need the traditional methods. The DNA isn't perfect yet, but we're hoping that this adds a new kind of toolbox, adds to the toolbox then of environmental DNA or biodiversity monitoring. That's the plan anyway. I'm sorry if I've gone over time. I was probably rambling. Thank you all for listening anyway. And yeah, if you need to contact me, feel free. And I will shut up now. So sorry.
Thanks, Molly. You certainly weren't rambling. Very detailed, very good overview of your research. If anyone has any questions, we do have one question from Donna. So at what level can this be developed? E.g., can freshwater macroinvertebrates be monitored using DNA to determine ecological quality of water? And the second part of the question is, how long will it stay in the environment? In other words, can you determine if it's living or dead historic samples of DNA? So at what level can it be developed?
So great question.
And the historic?
So we're hoping it can be developed a lot further. We've only used it currently for fish species. So we've only actually developed it for Atlantic salmon currently. I think in terms of monitoring ecological quality, I guess that would depend on our species and again, the databases. So I think the biodiversity monitoring and our meta-barcoding would be better for that in terms of monitoring quality of the ecology. Because you tend to look at a much broader picture than just a single species. But we're hoping for some to develop this system for Arctic char monitoring, because we know Arctic char is really sensitive to kind of these climate change and everything like that. So I guess it depends on the species you target for, but it's still kind of very new stages at the moment. In terms of how long the DNA stays in the environment, fantastic question. This again is a huge question within the environmental DNA space. So the problem with this and the problem with DNA monitoring on the whole is obviously all those other factors that are coming into place. So even just the water flow and how much, say, if you have a species upstream, is that just being washed downstream? Because the DNA can, depending on its state, so if it's in a cellular kind of state, it can last for quite a long time. And studies are still being done on this, so I wouldn't know exactly how long. And the kind of dead historic living, again, great question. You're asking all the questions that we're trying to answer in this field. So there's, I think a lot of studies at the moment have been like, look, DNA works. We can detect DNA in the environment. And we're now beginning to ask what type of DNA we're actually detecting, and kind of looking more at the ecology of the DNA itself. So I think at the moment, it's very much these questions actually are kind of unknown, and we're excited that you can detect it, but there's a lot of studies coming out now that are considering the effect of temperature and canopy kind of cover and UV light and everything like that is going to influence our DNA degradation, and kind of how reliable the system actually is. Yeah. Okay,
excellent. And actually, I just have a question myself, maybe. A few weeks ago, we had Peter Stewart at one of our ESAI webinars out in DCU, and he was talking about the whole health crisis and the link to biodiversity, and why the importance of biodiversity, particularly with pandemics. Mm-hmm. So I'm just wondering, it's a general question, can this method be used, for example, to look at things like COVID in the environment or other similar viruses that would be present through DNA? Or is anyone looking at that?
So there's no reason why it shouldn't be able to. It's actually an area that I'm hoping to get postdoc funding in, to kind of look into that. So at the moment, from our standpoint, we've only kind of looked at fish, but from the medical standpoint, the system that we've kind of adapted was initially developed for HPV detection from blood samples. So it is able to detect viruses and bacteria as well. They've both been kind of pathogens is kind of the main area of study in the medical field. So there's no reason that it shouldn't be able to also detect those systems in, say, the water body, assuming they're at a high enough level, because the issue then will be with our sensitivity of detection. But if they have nucleic acids and there's actually a Cas system that also detects RNA, so slightly different. So if the viruses are RNA based, we could just use a slightly different nuclease and slightly different design to also detect them. So I'm hopeful, but I've not seen anyone working on it yet. And I'm hoping in a year's time when I finish my PhD, someone will fund me to look at it. Yeah.
Excellent. And then just a final question to same one that we put to Philip, just in terms of if anyone out there is kind of thinking about applying for a postgraduate of the year, and maybe thinks they're not at quite at that stage, or they're talking about it. What advice would you have for them, how to go about it?
See, I'd agree. I'd say go for it. I was in kind of two minds myself, because I was only, when did I apply? Just starting my third year of my PhD when I applied. And I think you can get quite put off because you think of it being kind of a final year, someone has to have done all their research. But I think sometimes with some projects, I like to think I was lucky. I kind of had the breakthrough, as we might call it, fairly early on. And I've always tried to disseminate it in the best way that I can, because I think that's so hugely important in science anyway, that kind of dissemination part. I think it's very easy to get stuck in our lab and just talking amongst each other, but getting it out there to the people who we're trying to do the work from, I think is hugely important. And I'd say just go for it. I mean, I didn't expect to win it, I'm not going to lie to you, at all, because I was like, there are going to be people who are much further along through their research. But even going through the application made me really think of some, as kind of Philip was saying, some kind of huge questions that I needed to think about in my own research. So even just how you present your research, with the detailed kind of description that you have to give, which actually still isn't very long. It's quite you have to kind of really narrow down on what your research is and what impact it is going to have and why you're doing it, because I think I hadn't necessarily thought about the impact of it until I filled out the application. So I think even if you don't feel ready to go for it yet, as kind of Philip said, go through the questions, question your own research and question the impact you are having and how much you are getting it out there, and then it will change the way you think about it, and potentially then you will be ready kind of later in your research.
Good stuff, Molly. Thanks very much.
No worries.
And just to everyone, Molly will be presenting her work also at Environ, which is out in DCU, which will be through the virtual universe that we all live in now in October. And Philip also will be presenting further. So they can be contacted via email, or you can also engage if you have any questions about their work through ESAI and through Environ. So just finally, thanks to everyone for joining. Excellent presentations. So John, if you just want to run very quickly through some upcoming events.
Sure, thanks Liam. I'll just take 2 minutes and I'll let you maybe perhaps close out. So everyone, thank you for joining today. And if you are not quite zoomed out by the end of today, we're just trying to do a few more social engagement events where you can maybe interact with our council members in a more informal setting. So this evening we're having an environmental quiz of the year, hosted by one of our council members, Nate Power from CIT, and you're most welcome to join us, and you can just register via the website. The link is in our events, news and events section. But I suppose next week is our last webinar of this part of the summer series, although we're now in planning perhaps 1 or 2 before Environ, to kind of warm us all up for Environ 2020. But it's a very, very informative set of speakers looking at how they have dealt with adversity in research, specifically with dealing with COVID, and how some of us have now been working from home for several months and have had to adapt our our schedules, our agendas, our actual work programs. And so 3 speakers next Thursday will give their perspectives. One looking back during Foot and Mouth, which happened many years ago, Professor Enda Hayes from Bristol. But then we have an EPA and NUI Galway speakers, Carl O'Sullivan and Catherine Schonrock, excuse me, giving perspectives on what they have done and some advice on how you can refocus your energies during this pandemic. And also 2 other more social elements in your spare time before the end of the month. There's the Nature is Art competition. We'd love for you to submit an entry, a photograph that captures the natural environment, and the deadline's 31st of July, and also a Taskmaster challenge where you record a short video of your best environmental superhero, which is a bit more ambitious for our members. So that's it. Liam, I'll let you close out.
Thanks, John. So thanks very much, everyone, for joining. John's outlined a lot of ways to engage, but I would just close off by saying the best way to engage is join ESAI. Get on the website. If you're not a member, join now. If you know someone who's thinking about it, get on to them, get them to join. I think more important than ever is the importance of scientific research and the importance of communicating that. We see that day in, day out at different news conferences. So I think it's really important to have a vibrant scientific community, and in the ESAI we're trying to support and be a part of that. So thanks very much, everyone, and special thanks to Philip and Molly. I think the future is safe, looking at the quality of the research and presentation. The rest of us can retire. I include myself in that category. So thanks everyone for joining. Please engage over the next couple of weeks. Check out the web, and best of luck to everyone. Thanks.